After heroically getting, I think, to his office from his home following a hurricane, we have Mingshan to present our last talk in this session before we get to the panel discussion. Mingshan will present his work using various STX-BP1 mouse models. And the stage is yours, Mingshan.
Thank you, Scott. I will start sharing my screen. Really, first, I want to thank Scott and Tracy for giving me this opportunity to present some of the work that we have been doing on STX-BP1 related disorder. So this, most of the work that I will be presenting today were done by a number of people from my group, particularly the postdoc Charles, with some help from Joanne, Jack, and other people of the lab, right? So as we learned that there are many causes of LGS. So in fact, there are so many different causes that we have, but they all converge on some very common EEG phenotype, as we learned yesterday, even though they are still very heterogeneous. So our thought was that perhaps the genetic factor, even though they are only 15% of the total cause, could be something that we can model a little bit better in animal model, and perhaps we can have a better way to tackle the mechanism and also develop therapeutics for this disorder. So this is how we are thinking about this problem, and this is what I plan to share with the group, that how we use mouse model to model one of the gene that is a cause of LGS, and hopefully that we can learn from this exercise. So the other reason I want to share about this project is because personally this project was inspired by Caroline DeLuca, who I met when I was a postdoc at UCSD over 10 years ago. And she had this particular disease. So when I started my lab, I decided to work on this. At the time, it was considered really, really rare, but actually later on, it turns out STX-BP1 is actually one of the most frequent cause of pediatric epilepsy in humans, right? As you see in this list, it's definitely around, among the top 5 causes of the epilepsy. So in fact, mutation in STX-BP1 can cause many different types of syndromes, including Otohara syndrome, West syndrome, or infantile spasm, and as well as LGS and Dravet syndrome. And some very large-scale sequencing study has shown that, in fact, this gene is one of the most frequently mutated genes in sporadic developmental disorders. So because most of the mutation we have discovered so far, or more than 60% of those, are truncating mutation, therefore hyperinsufficiency is a very important mechanism, or perhaps is the major mechanism of this disease. So all these patients have intellectual disability, and vast majority of them have epilepsy. So the EEG patterns are quite heterogeneous among different patients, and some are focal, multifocal. Another two feature that you often see in patients are hypomistria or burst suppression patterns. And most of them also have motor deficits like ataxia, dystonia, tremor, and so on. And a subset of patients also develop psychiatric deficits such as autistic feature, hyperactivity, anxiety, aggression, and so on. So what is this particular gene, STX-BP1? STX-BP1 actually encodes syntacin binding protein 1, or commonly known as MUNC18-1. So it is one of the core components of the, the transmitter release machinery. The absence of this gene in many model organisms, including worms, flies, and mice, actually abolish neurotransmitter release and cause embryonic lethality. Whereas in human patient, right, remember it is the heterozygote mutation that actually caused the disease. So our approach to this problem has been that we wanted to establish mouse model of this disease, and then we use this animal model to develop and to understand the mechanism and also develop therapies, right? So for today's talk, I will be more focusing on the mechanism. So for that, we actually established 3 different mouse lines. So they are the following. The first one is a knockout first allele, which we insert a trapping cassette after exon 6. So essentially, this will behave as a knockout allele or like a null allele. But because this trapping cassette can be removed by the flipase, and therefore we can convert this to a flux allele in which the exon 7 is flanked by 2 LOX P sites. And then, of course, if we remove the exon 7, then we end up with a conventional knockout allele, the so-called TM1D allele, as shown in here. In this case, a premature stop codon will be created in exon 8. So here is the Western blot to show that indeed both TM1D and TM1A are null allele because there's no protein in the homozygote mutant, and protein is reduced by 50% in the heterozygote mutants. So therefore, the heterozygote mutant animal will be the hyperinsufficiency model for this disease, which we will be using to characterize those animals in both behavior, neurobehavior assay and EEG assay. So our characterization are guided by the human phenotype, right? For example, we show that this animal have impaired motor function. In this case, where you place the animal on this wire grid, so the animal will walk, right? Because they have to precisely place their paw on the wire, otherwise they will slip, right? So then we quantify how many times they may arrow and slip their foot, and then this is basically what the foot slip assay will measure, right? So in this graph, you will see that the black or the blue are the wild type animal from 2 different alleles, and the red and orange are the heterozygote mutant. The open circle are female, the closed circle are males. And this graph setting will be consistent throughout the talk. Sorry. So as you can see here, the mutant animal clearly are making a lot more arrows than the wild type controls. Another motor assay is this vertical pole test, where we put the animal on the top of the poles, and then they will have to turn around and come down to the bottom of the cage. Right? Clearly, it's taking longer for the mutant animal to complete this task. Therefore, both assay indicating an impaired motor function for these animals. So we also test their learning memory using the fear conditioning paradigm because all patients have intellectual disabilities. So in this type of experiment where we put the animal in a box and then play a song and then followed by a food shock, right? So this is an aversive experience. And then the next day, you put the animal back to the same box, but without the song, without the food shock. If the animal remember this is where it received the food shock, then it will freeze, right? So we use this freezing behavior as an indication of their memory. You can also put the animal into a different box, a different environment from where it received the shock yesterday, but you play the song. And again, if the animal can associate the song with the food shock as a memory, then it will freeze. Again, we use this freezing behavior to quantify their memories. So as you can see here, both the contextual and the cue memory are drastically impaired in the mutant animals as their freezing behavior are reduced very significantly. So the final characterization of this animal model is the long-term EEG monitoring, where we were interested to see if this animal have any abnormal EEG or develop any seizures. So for these experiments, we implant the electrode and record video EEG and EMG from free-moving animal for several days. We identified two types of abnormal EEG or epilepsy seizures. This include a spike wave discharge. It's typical of absence seizure. Another one is the myoclonic seizure, which I will show you in a minute. So first, you observe that this mutant animal show a lot of this spike wave discharge, as you see here. So on the left is a typical recording from the wild type animal. The traces are from the left frontal cortex, left somatosensory cortex, and the right somatosensory cortex. The EMG is from the neck muscle. So if you expand one of these spike wave discharge, you can see a typical spike wave patterns, which is around 5 to 7 hertz in rodent. In human, it's about 3 to 4 hertz. So we quantify the number of these events or the frequency of these events. We plotted data as a function of time in a day. And the wild type control data is on the left axis. The mutant data is on the right axis. There's 20 times the difference on the axis, right? As you can see here, clearly the mutant had several fold increase of SWD regardless the time of the day. But there's also an increase of these events in the evening. Another seizure type is the myoclonic seizure. Here is a video to show you what it looked like in these animal models. So again, this is, okay, now the video is playing. This vertical line indicating the current frame of the video. And you can see when there's spikes happen on in the cortex, you can clearly see the animal is making this myoclonic jerks, which can happen a number of times in a row, or it could also be isolated events. Another more dramatic version of this myoclonic seizure is, we call it myoclonic jump, right? So this is a mutant animal actually sleeping, right? So as you can see, it's sleeping. Suddenly, it will actually jump to the other side of the cage and then wake up from this, this traumatic events. And this behavior doesn't happen in the normal wild type animals. So here's the quantification, whereas the jerk happens in both. Some of these jerk are probably physiological jerk that we all experience. So therefore, you will see them in the wild type animal. But the mutant clearly have a lot more of those events. For the jump, so as I said, this doesn't happen in wild type animal, particularly during sleep, right? But this happen a lot in the mutant animals. So I hope so far that I have convinced you that we have created two different hyperinsufficiency mouse model that can recapitulate the key neurological phenotype of this disorder, including intellectual disability, epilepsy, and motor dysfunction. Now, with this model, the lab is working on two directions. One is to study the disease mechanism, like what cell type or what circuitry are critical. Another direction is to use this animal model to develop therapeutic strategies, like small molecule or genetic therapy and so on. So for the rest of my talk, I will continue on showing you some of the recent data on the mechanism of this disease. So a key question that we want to answer is that what cell type are critical for the pathogenesis of XDX-BP1. So because this will help us to understand the mechanism, but also it's important for us to identify the target for therapeutics, right? Because in order to know what will be the most critical cell type that we have to develop, we have to deliver our therapy to those cell types, right? If it's a gene therapy, then we need to know what cell type we must target, right? So for this experiment, we basically want to create cell type specific XDX-BP1 hyperinsufficiency model. So here's a cartoon to show you what the normal brain will look like, right? So this is, here I use red and the blue cell to represent excitatory and inhibitory neurons in the brain. And in the hyperinsufficiency model that I have shown you so far, every cell heterozygote for XDX-BP1, therefore, as shown here. And we want to create a glutamatergic hyperinsufficiency model or GABAergic insufficient model where the glutamatergic neuron will be heterozygote for XDX-BP1, where the rest of the cell are normal, right? Vice versa. In this case, the GABAergic cell will be heterozygote mutation for XDX-BP1, and other cells are normal. So to do this, we use the flux allele that we created earlier, right? And we cross to a VGAT CRE, which labels all GABAergic neuron in all GABAergic cell in mice. Or we can use a VGLU2 IRES CRE, which target all glutamatergic neuron during early development. Therefore, we can remove one copy of XDX-BP1 in all glutamatergic neurons. So from these two separate crosses, we can generate 6 different genotypes, right? Wild type, heterozygote flux allele, CRE alone, or the heterozygote conditional heterozygote mutants in either GABAergic cells or glutamatergic neurons, right? So now we compare these two type of heterozygote knockouts. And first, again, in the motor function assay, the foot slip or the vertical pull test, you immediately saw that the impairment only show up in the heterozygote, the VGAT heterozygote mutant animal, whereas the the glutamatergic heterozygote mutant are quite normal compared to all 3 control groups. So again, here, the open circle are female, the closed circle are male. The color match to the genotype down below here, right? So therefore, the GABAergic specific heterozygote animal recapitulate the motor dysfunction we saw in the constitutive heterozygote mutants. Similarly, when we look at their learning memory in the fear conditioning paradigms, we saw something quite interesting because the contextual memory and the cue memory deficit are actually segregated between these 2 cell types. So for the contextual memory, the VGAT heterozygote mutant animal show a strong impairment, but it's quite normal for the glutamatergic mutants. Whereas the cue memory, the sound-induced fear memory, it's impaired in the glutamatergic heterozygote mutants, but somewhat normal or actually slightly increased in the VGAT heterozygote mutants. So this is the first time that we actually see 2 aspects of the same same learning memory paradigm being segregated in 2 different cell types. So what about epilepsy, right? So the heterozygote mutant had 2 different epilepsies, spinal wave discharge, absence seizure, and myoclonic seizure. Again, very interesting that we saw that the SWD is only present in the glutamatergic heterozygote mutants, as you can see from this graph. The VGAT heterozygote mutants have basically no SWD, similar to all other controls. But when you look at the myoclonic seizure, for both the jerk and jump, they are clearly elevated in the VGAT conditional head, but does not present in the VGLU2 or glutamatergic conditional heterozygote mutants. Again, this is another interesting segregation of two different type of epilepsy being presented in two different cell types. So we actually have done many different other neurobehavior assays. So here's a general summary where both mutant animal have epilepsy, but they have different type of epilepsy. And the rest of the behavior are mostly recapitulated by the GABAergic specific heterozygote knockouts. As you can see here, the motor deficit is clearly shown in the VGAT heterozygotes, whereas additionally, the hyperactivity, the aggressive behavior, and the anxiety are all recapitulated in the VGAT conditional heads. And there's also an indication that the VGLU2 conditional knockouts show a small increase in the anxiety in one of those open field assays. So this data basically suggests that the GABAergic cells are particularly important for this disorder, even though the glutamatergic cells also make some very distinct contribution to the pathogenesis of this disorder. So then what would be the mechanism for the GABAergic contribution to this disorder? Like, what does this mutation actually do to the GABAergic synapses, right? Because as I said earlier, this is one of the core protein for neurotransmitter release. So in the mouse cortex, right, there are there's three major type of GABAergic inhibitory neuron, and two of those called parvalbumin interneuron or somatostatin interneuron are most well studied. So the parvalbumin interneuron consider about 40% of the total interneuron in the cortex. They tend to innervate the soma of the pyramidal cell, and they generate these fast spiking patterns, right? So very often people call them fast spiking interneurons. The somatostatin interneuron consider about 30% of the GABAergic cells, and they tend to innervate the distal dendrite, and they generate this regular spiking, right? So we wanted to know how does the heterozygote mutation affecting these two type of GABAergic inhibitory synapse in the cortex. So for this experiment, we perform pair recordings in brain slices where we record the postsynaptic neuron, the excitatory pyramidal cell as a postsynaptic neuron, and then use another electrode to record a parvalbumin interneuron and to evoke action potential, which is shown here. And then you can see these robust postsynaptic iPSCs, right? So, and when you do this for both the wild type or the heterozygote constituent knockouts, you observe that the connectivity or the probability for you to detect this connection are quite similar between the two genotypes, meaning the parvalbumin interneuron is still making the same number of connections to the postsynaptic targets in the mutant animal as the controls. However, the strength of this connection or the size of this unitary iPSCs is actually reduced about 30% in the mutant animals. As you can see, the average here, and this is the cumulative frequency as a function of the unitary iPSC amplitudes. When you look at the short-term dynamics of this synapse, right, by looking at how the synaptic response depressed over this 6 stimulus with a 100 millisecond interval, you can see the depression are quite similar. So therefore, overall, the postsynaptic neuron will receive less inhibition from the parvalbumin interneurons. Similarly, we performed the experiment with somatostatin interneuron. We observed something quite different. This time, we found that the connection probability from somatostatin interneuron to the postsynaptic pyramidal cell is actually reduced in the mutants. But if these interneurons are connected to the postsynaptic cells, then their connection strength is actually similar between these two genotypes, as shown here. Again, the short-term dynamics was not altered by these mutations. So this allows us to conclude that the glutamatergic and GABAergic neuron mediate two distinct disease features with very little overlap. Second, there must be two distinct mechanisms for this seizure in the hyperinsufficiency models, because each of the seizure type was recapitulated by one particular cell type specific deletion, but not the other. Third, and the GABAergic interneuron seems to be the most important cell type for this disorder, because the GABAergic hyperinsufficiency clearly recapitulate most of the phenotype that we observe in the constitutive heterozygote knockouts. And this data also reveal a possibility that we might be able to selectively modulate the disease phenotype by targeting one particular specific neuron transmitter system, right? For example, the GABAergic system. So to summarize my talk, I hope that I convinced you that we establish two genetic and construct valid model of XDX-BP1 encephalopathy. And now we are using this model to explore the therapeutic strategies. And we also show that the GABAergic synaptic dysfunction seems to be a critical contributor to the disease pathogenesis. And we want to know what mechanism underline the two distinct deficits of these two GABAergic interneuron types. Finally, as I showed, that the glutamatergic dysfunction seems to be very critical for the generation of the spike wave discharge, SWD. Therefore, we want to know how or what circuit mechanism is responsible for these seizure types. So to finish my talk, I just want to acknowledge the people who have contributed to the study, as I showed earlier, with additional help from my collaborators. And here's the funding source. So I'll stop here and be happy to take any questions.